for many of you, I know we're just moving the engineers now into the Cancer Institute. Many of you are not engineers, so you can maybe relate to that, but many of you are homeowners. So by the same notion, say you buy your new home, you don't have the wiring diagram for your house. You want to figure it out, but you got a big circuit breaker box in your basement. Right, so what do? You take your boom box, turn the volume up high, plug it in your kitchen, you run downstairs, you flip the switches till you hear the boom box go up, kitchen. You run back upstairs, go in the dining room, plug it in, and run downstairs, dining room. Right? You do that repeatedly, you get a first order network map of your house. That's what we realized you could do inside a cell. If you can make enough perturbations on your scheme, you could actually work out the structure of the network. And more importantly, not only the structure of the network, you could tell not only who's talking to whom, but what's the strength of the interaction and what's the nature of the interaction. Is it positive or is it inhibiting? We then thought about what could you do with this. And we realized that you could actually use this to address a very important problem in biotech, pharma, and cancer research, and that is use it to identify the targets of drugs. And how do you do that? Well, I teach control theory to our biomedical engineering undergraduates, and from lecture 1 to lecture 27, I emphasize that there's 3 parts to a control system. You have your input, your output, and the system itself. If you know 2 of the 3, you can work out the 3rd. Okay? So we realized that once you have your network structure, you could use this to filter the output of a drug, the output effect of a drug, and try to identify its targets. And the reason why this is so important is many of you can appreciate that we're in a target-rich and target-focused world right now. Pharma and biotech are really good at setting up assays to tell you what does the compound under development hit what it was designed to hit, right? What they can't tell you is what else might these things be hitting. And you have thousands of possibilities, right? And it's these other thousands of possibilities is why you'll have a drug fail or you'll get side effects. And we realized, okay, you know what? We can take those thousands of possibilities, filter it through the network, taking the output, and predict what was the likely input that actually produced this output. We've done this computationally. We've done this in bacteria. We've actually done it in mammalian. But just to give you a sense, I'm just going to tell you one very quick case study that we did a few years ago where we looked at fungal data, so yeast data. Here now we generalize it not just from using our switches. We actually found that we could use a range of perturbations to help infer the network. So we took 515 expression profiles that have been collected by Tim Hughes and his colleagues, used it to reconstruct on a genome-wide scale a network for yeast. From then this original really brilliant piece that Tim had published in Cell, he actually had 15 drugs that were profiled, 9 of which had no mode of action. So what we did was take each of those, pass it, take those expression data, take the output, and filter it through our reconstructed network. So what's shown here is this is the actual output for itraconazole, whose known target is ERG11. Okay? ERG11, in terms of expression profile alone, is buried down around 400 or 500 out of 6,000 possibilities. You would not pick it out. This is the line of significant differentially expressed genes. It's below that. Okay? You pass this output through our reconstructed genome scale network, and boom, ERG11 jumps up as number 2 predicted targets for that drug out of 6,000. When we sent this in, the reviewers said, okay, that's pretty interesting, but are you really going to hand a list of 50 or 100 targets to biotech or pharma? They're not going to like that. And it turned out we had about 50 here. And he says, why don't you try to do pathway enrichment, which is a really great suggestion. Because what you see here is in the yellow are actually the genes that are in the same pathway as ERG11, kind of all over the place. But when you pass them through, the top mediators being predicted, the identified targets, predicted targets through our approach, of the top 57 of them are in the same pathway as ERG11. So it now can nicely simplify the way you could look at this. For itraconazole, the known pathway that it targets ergosterol biosynthesis, known target is ERG11. From our analysis now through the network, our highly enriched pathway with steroid metabolism, of which 7 of the top 50 are linked with that, including as number 1, the top target and known target for itraconazole. And we basically did similarly well on the other 8 drugs. Hydroxyurea, known to hit DNA replication. Here's its 2 targets. This was the enriched pathway we picked up. And of the top 50, there were 4 predicted targets, 2 of which are the actual known targets. Second thing the reviewers said was, that's really kind of cool, seems like a new technology. We haven't learned anything from what you showed us. Right? You're looking at drugs of no mode of action. Can you go beyond that? And it was a really good challenge. And so Tim Gardner ran down the street, went to our chemistry department, and says, guys, we got a challenge here. And he talked to Sean Elliott and Scott Schaus and said, do you guys have anything interesting in your chemical library screen that is doing something funky in yeast? And they says, yeah, we've got this drug or compound PTSB, and it inhibits growth in yeast, but it also inhibits growth in lung cancer cells. And Tim said, great. And they said, hey, we don't know anything. We don't know what this thing is doing. And he says, drop it on a bunch of yeast, do an array, and give us the array. So they did that. Again, pathway that it hits and target were unknown. We predicted it would hit cell redox homeostasis and 2 targets, thyroreduction and thyroreductase. Scott Shao, Sean Aylett went in their labs, they actually had assays for that and actually showed that PTSB, in fact, did inhibit thyroidexin and thyroreductase. Probably hits other things, but we were able to go and show this. So here now you take a computational systems biology approach where you can take a large set of expression data and now take a drug of unknown mode of action and pass it through that to make an in silico prediction that turned out to be validated in vitro. This approach, as said, is now amenable really to cancer drugs, and we're very open to collaboration to see how you can do. We then, in the last 2 case studies, got excited to think about, okay, the perturbation need not be a drug, right? Basically saying, okay, you've got an input that could be an environmental perturbation, it could be a biological process, it could be a disease. And with Isla Aragon and a few others in our lab, we realized, boy, wouldn't it be neat if you could think about now passing a disease profile through your reconstructed network to begin to gain insight who are your key genetic mediators, both at the gene level and at the pathway level, underlying the development of a disease and or the state of the patient. And so Isla took that challenge up, and what she did was take about 1,200 different expression profiles across many different cancers, use those to reconstruct a quantitative human network, and then looked at non-recurrent primary prostate cancer samples from humans, as well as metastatic prostate cancer samples, passed them through our networks to see if she could identify the come up with predicted mediators, to try to zoom us in, right? And many of you can appreciate, if you look at expression data for microarrays, you've got hundreds, if not thousands of things changed. It gets very frustrating, right? And you'll see papers published, these 500 went up, these 700 went down. It's not really that insightful. But we thought, boy, if you could pass it through, could you maybe filter the process and have the key guys pop up? And to kind of focus ourselves, we hypothesized, it's been known that androgen receptor signaling, the AR signaling pathway, is a key guy underlying metastatic prostate cancer. So we hypothesized that this would be the key mediator for metastatic and that you would not see it from expression change alone. And in fact, that's in short what we found. So up here is the non-recurrent primary prostate cancer. If you look at expression change alone, the only enriched pathway was proteolysis, peptidolysis. If you passed it through our network analysis, metabolism came up, which is nice, right? You have a lot of interesting things about metabolic shifts in cancer, and even in the early stages. In the metastatic, if you did expression change alone, we had muscle development and muscle contraction with the key enriched pathways, and cell adhesion, so that's a pretty nice one. When you pass this expression profile through our network, the #1 enriched pathway with very high level of significance was the AR signaling pathway, which again was kind of a nice validation. It didn't come up with a new insight, but it did show that you could now use these network analyses to begin to gain insight into mediating aspects of pathways. The other thing we got excited about, and Daniel gave a great talk touching upon some of this notion, is could you use network analyses to get after biomarkers? My wife's a primary care physician at Mass General who studies prostate cancer research. She's actually known as Queen of Prostate at the NIH. I'm not sure it's a great nickname to have. What she often tells us is that many men will die with prostate cancer, not of prostate cancer. And the challenge with many of these screens is, can you discriminate between the kitty cat tumor that's not going to do any harm, and it'd be better to just leave it there because the procedures have some ill-toward effects, versus the tigers, the tumors you want to get out. And we just, as a first step, moving towards kind of network-based markers, said, okay, if we look at the AR gene, could you now use the network to pull it up, pull it out of your scheme to use it as a marker? And what Isla did was actually now pull in recurrent prostate cancer data and compare those with non-recurrent and metastatic. And the AR gene, when you pass it through the network, is only about 843 out of, in this case, I think we looked at 22,000 possibilities. Expression change alone is about 600, not above at the level of significance. There is no difference when you look at expression change alone only between the non-recurrent and the current, but it pops up into the level of significance when you pass it through the network. So now it becomes a significant measure when you filter it through your network. And then when you look at the metastatic case, it pops up to a very high level, and it's still below the measure of significance on expression change alone. Very last quick case study, and it speaks to a number of collaborations I have with folks in this room, is we also got excited about, could you use these network analyses to get after combination therapies? We were challenged by Ron DePino and Linda Chin and Bob Langer and Bill Hahn to think about, could these systems approaches help you identify who do you want to hit? And could you hit them with RNAi or small molecules? So the general motivation now was, okay, if we could recreate your network, reconstruct it, filter a different disease state, can you now identify mediators you want to hit, as well as can you identify certain disease defense pathways that you might want to knock down to get a better response, which now could identify your key guys for combination therapy. What we did with input from Ron and Linda and others was then look at HCC liver cancer So we looked at expression data from patients as well as controls to see could we come up with mediators. Firstly, just looking at the predicted genetic mediators. We didn't know anything about this disease, which has its advantages in that we truly are objective, disadvantages in that we don't really know what to make of the output. When we came up with our list of mediators, 200, 300, we didn't really know what to make, and we went over to Ron DePino, and Ron was great, and he looked at it and he says, oh, you've got the greatest hits list here. You've got a lot of interesting genes, and with his input, we went and focused on several, and what you see here, these are the rankings out of the 22,000 of predicted mediators coming through the network, and here they are on expression change alone. So many of the interesting ones we came up with, like HU1, you would never have picked up. It's down around 4,000. It's well in the noise of expression change. But that change that it exhibits, when you reflect that it sits in a network, turned out to be very important. We're saying it's actually the eighth guy of driving much of the response that you see. We then can do pathway enrichment, and a number of these come up with interesting schemes linked to cancer cell cycle as well as cancers. And again, with Ron and Linda's input, we did a kind of a multi-pathway analysis and said, okay, here's two interesting mediating targets we're predicting to come up that seem to be impacting a lot of interesting pathways. And with Tara Deans, we then did some prelim analyses where we did RNAi knockdown on these guys, where the targets by themselves actually had a pretty big effect, but we got an even bigger and stronger effect when we did them together. And the challenge there is that we're obviously using the great expertise and insights of Linda and Ron. Could you do this objectively? And so with Zheng Li and Isla Aragon in our lab, we actually came up with a nonlinear analysis where you can now look at these network outputs and begin to make predictions on pairs or triplets or quadruplets or 5, 6, 7 number targets. You know, the guy like Bob Langer who can do magic on delivering schemes, you really don't have great limitation on the number of guys you could target. And what we then said was, could we objectively identify pairs to start out with of targets that seem together to have synergistic effects that if you could knock down, you could begin to get some disease reversal. And again, for HCC, the analysis predicted PRs, these two guys, whatever they are. And on their own, they did very little. But together, we actually got a dramatic and very big effect, again, which was all done objectively and came in for the analysis. We're going in many different directions from this. We're now working with Linda and Ron on glioblastoma. We're beginning to look at ovarian cancer. We're doing breast cancer work with Don Ingber. And again, I'm just so happy that with this new Koch Institute that the engineers are going to be in with the biologists. I think it's going to be a very exciting time in the field. Much of our stuff is published. I encourage you to visit. And this notion of kind of engineering circuits, will it revolutionize medicine, engineering, biotech? I'm not so sure. Let's take out engineering life. Let's just say engineering. I think engineering in conjunction with biology will really have a big impact in these next few decades. And I think, obviously, the Koch Institute is going to play a big role. Thank you, everyone.